This is podcast 22, continuing our discussion about how DNA is packed in the cell and its composition. When you look at a chromosome, the fine structure, we talked about how the nucleosome is arranged with the 8 histone units and then the one H1 histone unit off to the side there. But on a macro scale, you can see a constricted area of a chromosome called the centromere, and it doesn't have to be in, it can vary along its position along the chromosome, and at the ends of the chromosomes there are telomeres, and these are regions of highly repetitive DNA. And in fact, highly repetitive DNA is most of your DNA. So in classes like this, we talk mostly about the process of DNA replication and then transcription and then translation. And we talk about it in the context of how DNA gets transcribed to RNA, which gets translated into protein. That's really true for only 1.5% of your genome. So most of the DNA that you would see in a nucleus is not coding for protein, or indeed coding for genes that code for ribosomal RNA or transfer RNA. Instead, most of your DNA is what's called repeated DNA. So these are stretches of DNA sequence, let's say AGGTT or something like that, that's just repeated over and over and over again. They can be repeated back to back, like AGGTT and then AGGTT again, thousands of those in a row. That would be tandem repeats. We could be talking about introns, which are part of a coded gene, but not that information is cut out when the mRNA is transcribed and processed. After it's transcribed and processed, in that processing, the introns get cut out. Okay, and then fully half, a little over half of your DNA is just repeated DNA that is repeated in different spots throughout your genome in different chromosomes, including what are commonly called ALU elements because they respond to an restriction enzyme called ALU. So all of this gets transcribed at some time during the lifetime of a cell, but most of what we think about as being something being transcribed and then turning into a protein, that's a very small percentage of the pie. A fair bit of what gets transcribed gets cut out before it even leaves the nucleus in the form of a neutron. A fair bit of your genome is made up of a repeated element that is repeated over and over and over again. And then there's some non-coding DNA, does not code for RNA or protein, that is, and it doesn't have a repeated sequence, so it's unique sequence, but we don't know necessarily its function. We used to call this part of the pie, everything except for this deep blue, we used to call this junk DNA because we did not know what it did. There was a big project done about 5 years ago that showed that all of this is transcribed at some time during your life, so it's likely that we just don't know yet what the function of all these other elements of your genome are, but maybe by the time you're teaching this class we will know. Okay, if you look at a nucleus in a transmission electron micrograph, it'll stain. Uranyl acetate is a typical stain, or lead is another typical stain that is used in electron micrographs, and different parts of the nucleus will stain darker or not as dark. So you have nucleolus, which has high concentration of RNA. You have euchromatin, this lighter color stuff, and heterochromatin, and then we'll talk more about nuclear pores in a bit. So the euchromatin has a looser structure and is more actively transcribed. The heterochromatin is more compact and is not currently being transcribed at the point in which this image or this cell was fixed to make this image. But heterochromatin can become euchromatin, euchromatin can become heterochromatin, depending on when you're looking at the cell. So euchromatin, a looser structure, is needed in order to transcribe that DNA. If the DNA is very tightly wound, then it can't be transcribed. And as we saw in the last podcast, that is one way to regulate what genes you're accessing. Is you can change the modifications on the histone proteins on which the DNA is wound, and by changing the histone structure, by modifying that histone structure with methylation or acetylation, you change the tightness at which the DNA is wound around it, and you change its ability to be transcribed, which changes gene expression. Okay. If you see this sort of picture, these are chromosomes like you have seen in previous classes, and these are the sorts of chromosomes that you would see dragged around during mitosis and meiosis. This is what it would look like when the image was taken, and then the computer will separate out the homologous pairs. So it's the first number 1 chromosome, pair it with the second number 1 chromosome, even though they might be on different parts of the original smear. So this is computer generated. They're not all lined up like this in real life. But these are chromosomes when they're condensed and the cell is getting ready to divide. When the cell is not getting ready to divide, it's just living its life as a cell, such as in the G1 or G0 phase, the DNA is less condensed, not looking like this, but more spread out in heterochromatin and euchromatin. And even in that state, it is intact as a linear molecule. So, for example, one copy of chromosome 11 is all in this area. It's not like you have part of chromosome 11 here and part of chromosome 11 over here. Here is one copy of chromosome 1, and then here's the homologous chromosome 1, each one intact in its area, and not next to each other, but as a one linear molecule all together. So this is the discussion in the last podcast from the video that said chromosomes don't appear until this part of the cell cycle. They're still chromosomes as far as molecular biologists are concerned, even in euchromatin and heterochromatin, in that they are one structure, but they're more spread out. This technique, where you can color things like features like this on DNA, is called FISH, or fluorescence in situ hybridization, where you have a fluorescent tag that has a color that a microscope can see, and that fluorescent tag is specific for a specific sequence. So then you have different colors for different sequences. You know what sequences are present on, say, chromosome 5, and then you put that on a tag that is this color green, and then you see every place where chromosome 5 is with that color green. Okay. FISH can be more precise than this too. Like if you're looking for a particular gene on a chromosome, you can get like a band of the chromosome lining up. Okay, mitochondria have chromosomes too. They are circular, just like prokaryotes. And just like prokaryotes, almost all of the chromosome codes for something. So there's a smaller number of genes, 37 genes versus, say, 23,000 in the nuclear genome of humans. But there are no introns and a very small amount of non-coding DNA, unlike in the nuclear genome, which most of it is non-coding for protein DNA. So if you do a map of the mitochondrial chromosome, you can see where different things are coded for, including different parts of the electron transport chain. A lot of mitochondrial genes cooperate, in quotes, with nuclear genes in that they are a multi-subunit complex, and some of those subunits are coded for by the mitochondrial genome and then translated on mitochondrial ribosomes and reside in the mitochondria. Some of the subunits would be coded for by the nuclear genome. They are then processed and translated on endoplasmic reticulum ribosomes, and then they have a tag for import into the mitochondrion for final assembly of, say, cytochrome oxidase. So cytochrome oxidase is one of my favorite examples because there's 13 subunits. 3 of them are coded for by this thing, the mitochondrial genome. 10 are coded for by the nuclear genome, and they all have to arrive on the intermitochondrial membrane in order for eukaryotic life to proceed. Okay, so mitochondria does have a genome. It's not really fair to say how many copies of the mitochondrial genome there are per mitochondrion, because remember, the mitochondrion is this large anastomosing structure within the cell, is constantly changing. So there are, if you were able to assay all the mitochondrial volume in the cell, there will be several copies, several hundred copies, if not thousands of copies of the mitochondrial genome within that big network of mitochondria. Okay, H5P question for you. Okay, moving on to the nucleus, where the genome is going to reside, except for the mitochondrial genome. The nuclear genome obviously resides in the nucleus. Nucleus has a very interesting structure in that there are hundreds to thousands of nuclear pore complexes that transverse the nuclear envelope, and then the nuclear envelope is a double membrane system with space in between the two membranes called the perinuclear space, and that is part of the endoplasmic reticulum. The rough endoplasmic reticulum and smooth endoplasmic reticulum is contiguous with the nuclear pore. So this is how it's drawn and how it should be drawn. If I ask you to draw it on a test, you don't want to draw this as a hard line. You want to see that this is connected to and becomes the ER. In a freeze fracture, you can see nuclear pores really well. The outer surface here is of the envelope is seen in like the skin sort of thing, and you see the outer nuclear pore, outer shape of the nuclear pore, and then showing the inner surface, you see also an indentation as part of the nuclear pore. And if we zoom in on a nuclear pore, it's a very complex protein structure that has a lot of structural elements to it. It's arranged sort of like a wheel that's embedded in the nuclear envelope. There are anchor proteins that attach it to the envelope. There are spokes that maintain it as a structure. There's a basket on the nuclear side of this. There is a central element called a transporter that probably contributes to how things are passively moving in and out of the nucleus. And in general, if you are less than about 30,000 molecular weight, it also depends on your shape. But in general, if you're less than 30,000 Daltons in molecular weight, then you can move through the nuclear pore, probably through this transporter complex. The size of this hole for the nuclear pore is lots bigger than this. So you would predict that things that are bigger than this can get through the nuclear pore, but they don't without a special transport system. And so that's why it's thought that there's some sort of filter sort of effect within the nuclear pore, probably associated with this transporter, that says if you're bigger than about 30,000 Daltons in molecular weight, you're not going to just travel through the nuclear pore. If you are bigger, it doesn't mean you can't get into the nucleus. It just means that you need a nuclear localization signal, NLS, and which is an amino acid tag that's between 8 and 30 amino acids long. This is part of the transcript that would be coded for by the nucleus, and then that transcript is processed, and then it has to leave through a nuclear pore, and then gets translated on a ribosome, and then the resulting protein would have to get back into the nucleus through a nuclear localization signal. So it's coding for its own return trip, so to speak. So one of the things I might ask you on an extended response is to think about all the things that have to get in and get out of a nucleus for a nucleus to do its job. So one advantage to having your genome inside a nucleus is that you can transcribe things, and if it's a long thing you're transcribing, you can be sure that it's not going to get translated by a ribosome, which is outside the nucleus, until you're done processing it. So you cut out all the introns, you put together the exons that you want to put together, and now it's ready to leave the nucleus and be translated. So that's an advantage. But a disadvantage is all the machinery to do that sort of thing has to be made outside the nucleus and then brought into the nucleus. And so I want you to think about all the stuff that has got to get in and get out of the nucleus for the cell to do its job. Now I'm going to read you a paragraph from the book that gives you the sort of things that I'm thinking about, and you may be surprised about some of these numbers. Okay. To get some idea of how much traffic must travel through the nuclear pores, consider the flow of ribosomal subunits from the nucleus to the cytosol. So, of course, ribosomes are only one thing, but still an impressive thing. Okay, ribosomes are partially assembled in the nucleus as two classes of subunits, each of which is a complex of RNA and proteins. These subunits move to the cytosol, and when needed for protein synthesis, are combined into functional ribosomes containing one of each type of subunit, large and small. An actively growing mammalian cell can be synthesizing 20,000 ribosomal subunits per minute. We already know that a cell has about 3,000 or 4,000 nuclear pores, so lots of nuclear pores, 3,000 or 4,000 per nucleus. So ribosomal subunits must be transported to the cytosol at a rate of about 5 to 6 subunits per minute per pore. Okay, just doing the division there. So traffic in the opposite direction is, if anything, even heavier. When chromosomes are being replicated, okay, cell is getting ready to do cell division, histones are needed at a rate of about 300,000 molecules per minute. Okay, so you're doubling your DNA. You've got to wrap all that DNA around histones, and you need about 300,000 histone molecules per minute. The rate of inward movement must therefore be about 100 histone molecules per minute per pore. So each of these pores, every minute, is moving in about 100 histone molecules and 5 or 6 ribosomal subunits are moving out at the same time. And that's just to consider those two things. And there's lots of other things that nuclei do. So in addition to all this macromolecular traffic, the pores mediate the transport of smaller particles, molecules, and ions. So I want you to think about that. You can think about it with your study partners, with your group, and then think about how you would answer an extended response like that. If I didn't just give you, and if I said you can't use the example that's in the podcast, what would you say? Okay, so let's think about some of the things that have to go in and out of a nucleus. All right, so DNA is being transcribed and then processed. So ribosomal RNA is going to be coming out of the nucleus to make ribosomes. Messenger RNA is going to be coming out of the nucleus. And transfer RNAs are going to be coming out of the nucleus. Okay? Those RNAs are also probably attached to proteins and not just naked RNA going through. So that makes them a little bit bigger. mRNAs are also being translated on ribosomes in the ER and in the cytoplasm, and they have to get back into the nucleus because the nucleus has jobs to do like we just talked about, like duplicating its DNA, or proteins that are part of transcription. We're going to be talking about that soon, and you're going to see there's just gobs and gobs of protein involved in the process for transcription. Okay? So, again, think about all the stuff that's got to get in and out of the nucleus through those nuclear pores. All right, so if you're bigger than about 30,000 Daltons, how do you get in? You need a nuclear localization signal, NLS, on this protein, and this is part of what gets translated, this nuclear localization signal. It binds to a protein called importin, not important, but importin, okay? Then the importin and its cargo go through the nuclear pore complex, okay? And once they're in the nucleoplasm, okay, the complex binds to a protein called RAN, which is in the nucleus, plus GTP, okay? And then that GTP is split to GDP by GEF. You're going to see GF again. GEF stands for guanine nucleotide exchange factor. GTP gets switched out for GDP and is split there. And then that the cargo is released into the nucleus. To get the importin back out into the cytoplasm, so it can do this again, the GTP plus RAN goes back out through the nuclear pore complex. Okay, now it is here bound to importin, and GAP, GTPase activating protein, splits the GTP into GDP plus P and releases importin. Okay, so I said before that GEF splits the GTP. That's not technically true. GEF is just switching out GTP for GDP, whereas GAP, this guy here, is actually splitting the GTP. So to go over this again, you want to get, you're bigger than $30,000, or $30,000 Daltons, and you want to get into the nucleus. You have a nuclear localization signal. It binds to this protein importin. Importin plus the cargo goes into the nuclear pore complex. Nuclear pore complex on the nucleoplasm side binds to RAN plus GTP. And that will take off the importin and switch it on to the RAN protein, releasing the cargo protein. And then the RAN also at the same time gets its GTP switched out for GDP by GEF. Then GTP plus RAN plus importin now goes back out the nucleus into the cytoplasm to return importin. And to get importin off of RAN, then GTP is split by GAP, GTP activating protein. That releases importin, so it can do this again. And now you have RAN plus GDP outside in the cytoplasm. Okay, H5P question for you.